Start off, let's differentiate between the term transcribe and translate. Transcribe is when you copy from one medium to another, whereas translate is when you convert into a different language. When you look at the DNA, when the information in DNA is copied into mRNA, that is called transcription. In the process of translation, ribosomes use mRNA to synthesize protein that contains a specific series of amino acids. The process of translation involves 3 steps. These 3 steps are similar in transcription. These involve initiation, elongation, and termination. Initiation is starting off, elongation is addition to the protein chain, and termination is finishing up the process. In the process of initiation, a complex of small ribosomal subunit and the initiator tRNA binds to the 5' cap. This complex starts by scanning from the 5' end to find the AUG, which is the start codon. The methionine is then brought to the start codon AUG by the methionine tRNA, which is the initiator tRNA. The large ribosomal subunit joins to form the initiation complex. The ribosomal assembles so that the start codon is in the P site. This is called the translational initiation complex. So after the initiation complex is formed, but before any amino acid have been linked to make a chain, our first methionine carrying the tRNA starts in the middle slot of the ribosome where the P site is. Next to it is the fresh codon, which is exposed in the another slot at the A site. The A site will be the landing site for the next tRNA, whose anticodon is complementary match for the exposed codon. Once the tRNA has landed in the A site, after this is when the peptide bond formation occurs, which connects one amino acid to another. This step transfers the methionine, which was right here, from this tRNA to the next tRNA in the A site onto the next tRNA's amino acid. Once the peptide bond is formed, the mRNA is shifted, so it is pulled onward through the ribosome by exactly one codon. This shift from here to here, this shift allows the empty tRNA to exit from the E site. So this will now exit from here. And it also exposes a new codon in the A site, so the whole cycle can repeat again. In the step of elongation, the 3 sites P, A, and E are very important. The P site is where the first tRNA was brought to at the start, AUG, at the start codon, and the A site where the second tRNA entered and the addition of amino acid at the A site occurred. E site was where the exit for the empty tRNA. Elongation leads us to the next step of termination in the process of translation. In termination, a stop codon, which is either UAG, UAA, or UGA, is recognized by the release factor in the A site. When a stop codon is encountered, a release factor binds to the A site. the polypeptide chain is released, and the ribosome in the very last step will disassemble. As the ribosome disassembles in the very last step, a newly made protein will be released. In short, the termination is basically a release factor protein that helps in dismantling of the ribosome mRNA complex and releasing the polypeptide chain. To summarize the process of translation, translation is the process of synthesizing protein. It is divided into 3 steps: initiation, elongation, and termination. Initiation starts off the process. It is when the mRNA enters ribosome. Elongation is the addition to the protein chain. It is when the ribosome reads one mRNA codon at a time. Termination is the very last step in the process of translation, which is finishing up, and it is when tRNA delivers amino acids until a protein is created.